Hello, everyone, and welcome to today's webinar titled Innovative Genomics and Cold Chain Solutions for Development of Cell and Gene Therapies. I'm Susie Valdez of LabRoots, and I will be your moderator for today's presentation. I'd like to invite everyone to participate by submitting your questions during the presentation. To do so, simply type them into the Ask a Question box and click Send. And if you have any trouble seeing or hearing this presentation, just click on that question mark icon on the left toolbar for further help and submit your concerns to the Ask a Question box. Now let's get started. Today's webinar will be presented by Dr. Elizabeth Louie, Supervisor of Technical Applications at GENEWIZ, a Brook Life Science company. And in her role at GENEWIZ, Dr. Louie provides consultation to customers across the GENEWIZ genomics solution, including next-generation sequencing, gene and DNA synthesis, and any custom molecular biology need. For a complete biography of Dr. Elizabeth Louie, please click on the Biography tab at the top of your screen. Dr. Louie, welcome. You may now begin your presentation.
Thank you for the introduction, and thank you so much for giving me the opportunity to speak about some of the exciting solutions we have in the cell and gene therapy space here at GENEWIZ and at Brook Life Sciences. So first, I wanted to take a quick look at the topics that we'll be covering. We'll first start with an overview of our capabilities, then we'll perform a spotlight on processes for AAV-based therapies, and then I will talk about our expertise in the biomaterials management within the R&D space, our expertise in clinical trial support, and also highlight our benefits of the comprehensive supply chain solutions that we offer. Here, we've described the general therapy development pipeline and the individual stages where some of our offerings will be applied. From screening and identification to preclinical testing and manufacturing, and throughout clinical trials and therapy delivery, Brooks and GENEWIZ has many solutions to elevate and support your program from discovery to distribution. So let's jump right in to some of our genomic solutions. This is a closer look at the previous breakdown. Here at GENEWIZ, we have options for analytical services under our next generation sequencing and Sanger-based products, as well as synthetic DNA offerings. Our research-grade offerings are supplemented by both GLP-compliant and CLIA-validated options, and we have several proprietary protocols developed that specifically target the needs of cell and gene therapy researchers. To highlight our capabilities, I want to take a deeper dive into some of these proprietary protocols for AAV development. So for those who are not familiar, AAV, or also known as adeno-associated viruses, is a single-stranded DNA virus genome. It has two inverted tandem repeats, or ITRs, that are at both ends of the genome and flank the critical genes that are needed for viral replication and encapsulation. This encapsulation is necessary as the delivery vehicle and is required when being introduced into a host or a patient. Now, in recombinant AAV production for therapy, 3 plasmids containing those critical replication and encapsulation genes, as well as that gene product that's going to be delivered, are transfected into a packaged cell line. That cell line then produces the AAV particles that will be used for the therapy. The transfer plasmid contains the aforementioned ITRs. Previous studies have shown that maintaining the ITR throughout the entire production pipeline from the plasmid to particle affects the quality and efficiency of the produced virus. Therefore, any errors to the ITR, if left undetected, would reduce reproducibility of experiments and can cause delays in translating its use into the clinical space. So ITRs are notoriously difficult to sequence through due to the inverted repeat containing a hairpin secondary structure. Further, historical limitations to sequencing hindered high-quality sequencing options to confirm packaged genomic DNA. By addressing constraints at the recombinant plasmid DNA level at the top, as well as the AAV virus particle stage shown on the bottom, our solutions ensure a good start for high-quality AAV manufacturing. So for the first stage of generating the plasmid DNA, we have a plasmid preparation and synthesis workflow that is designed to help confirm, maintain, and correct ITR. First, using our proprietary AAV-ITR sequencing protocol, we can sequence through the ITR hairpins with high quality. This allows us to determine if the ITR is intact, untruncated, or mutated. Depending on the results, GENEWIZ can then help correct, synthesize, and propagate that plasmid. To highlight the benefits of the protocols, I'd like to first show what the typical sequencing read looks like when sequencing across an ITR region. The start of the ITR hairpin in the chromatogram is underlined in red. As you can see, once the primer reaches the hairpin, there's a complete drop-off in the signal. In comparison, our AAV-ITR Sanger method can read through the entirety of the ITR, which is shown in that black box. And the read is good quality and is maintained throughout the other side. For any researchers using AAV libraries or researchers interested in determining what an individual ITR is in a mixed population, the next generation sequencing on the PacBio is a great option. The PacBio provides single molecule resolution for the entire plasmid at high accuracy. Its chemistry is known to sequence through GC-rich regions and the ITRs at high quality. And this workflow allows for high throughput types of screening and analyses, where the Sanger method is less feasible. So why is sequencing so important? There are traditional ITR confirmation protocols using digestion analysis, which is really considered the gold standard. In this case study, we performed an enzyme digestion of a plasmid product that will cut the ITRs and result in two bands. The bands are that gene of interest and the remaining backbone. If the right size bands are observed, the traditional assumption is that the ITRs were therefore present and intact and is released from QC. However, when we performed Sanger sequencing across the expected ITR regions, we found that one of the arms had been deleted, but not the arm that contained the enzyme binding site. Therefore, while digestion analysis provides a good look at any major deletions in one of the arms, it does not have the level of resolution that sequencing can provide. Therefore, it's important that sequencing should become part of a standard QC process. Now, confirming ITR is only half the issue. Now that we can confirm it, we can address the ways to maintain it. Now again, ITRs are known to mutate, and researchers spend many resources in screening and testing to get that right product. At GENEWIZ, we have a proprietary DNA plasmid preparation protocol that maintains the ITR better compared to existing commercial lines and protocols. In this case, we transformed 8 different AAV plasmids into several strains, and then we took the colonies from those strains and screened clones from each plate using our Sanger method. We found that the GENEWIZ protocols were best at keeping the ITR intact, regardless of which plasmid we are working with. For the other commercial strains and methods, some fared better than others, but the GENEWIZ method consistently came out on top. Of note, our protocols have a high success rate when generating large-scale DNA preparations, such as MIDI, mega, and gigascales. 98% of the large-scale DNA preparations that we tested maintained the ITR with the first propagation attempt, while the remaining 2%, we were able to get the correct ITR after a second attempt. For manufacturing, we have GLP-grade plasmid sequence confirmation services and also GLP DNA preparation services available. Our services are compliant with the FDA's 21 CFR Part 58 guidelines. We can perform a fully GLP-grade amplification and or digestion analysis for ITR verification. Our Sanger AAV ITR protocol is available as a supplement at the research grade level, though we expect to formally launch the GLP option in May 2021. So here, we've addressed the issues of ITR confirmation and maintenance at the plasmid level. So I'd now like to share the solutions that we have at the confirmation of the packaged virus. When it comes to the product that's the basis of your therapy, it's critical to understand what is being packaged. Impurities can include defective or truncated particles, residual DNA from other viruses, or contaminants from the original plasmid and the packaging cell line. Now, any impurities or incorrect sequences can trigger unwanted immune responses. Therefore, measuring the level of any DNA contaminant is important in understanding the risks of adverse side effects during clinical use. So we actually used next-generation sequencing to address these concerns. First, we confirm the packaged virus via Illumina sequencing, and we confirm the purity of the virus. This method provides deep sequencing of any DNA that can be packaged. The resulting data can be used to screen against other viral databases, against the host, and against the plasmid sequences to determine the ID. On a secondary level, the Illumina platform provides some insight into the mutations and truncations that would also occur in the virus itself. However, there are some limitations here, as the Illumina platform requires the genome to be fragmented prior to sequencing. Therefore, the data that's being generated for variants and deletions would be performed on the entire population level. For a deeper look into the exact nature of the sample, then sequence confirmation on the PacBio is a more ideal situation. This allows to screen the AAV without fragmentation, and therefore the sequences of any potentially mixed genome sample will be kept intact, and every individual sequence will be analyzed. This will provide physical confirmation of those truncations, those variants, and the ITRs at the single molecule level, avoiding the need of any inference or sequence assembly. Further, next-generation sequencing can be used to profile the patient or host microenvironment. We can perform immune response profiling and gene expression profiling during and after the viral infection or after a treatment. We can further screen for whole transcripts using long-read sequencing if the treatment affects expression of specific isoforms. Additionally, we can perform characterization of the AAV within the host and actually characterize the host itself using any DNA sequencing options. Now, all of these services are performed at the research grade level, but CLIA-validated and CLIA environment processing is also available when required. For AAV manufacturing, we do have GLP-grade viral or cell bank confirmation services available. This can be performed using our PCR-based Sanger sequencing pipeline. We accept a variety of materials, which are listed here, and all the work is performed compliant to FDA guidelines. So now I'd like to move out of our analytical workflows and want to talk more about our capabilities with Biomaterials Management in relation to the cell and gene therapy research and development space. So circling back to that original overview, Brooks Life Sciences has expertise in providing consumables, instruments, cryogenic solutions, and sample and material storage options throughout the entire therapy development pipeline. Our unique capabilities provides traceability, automation, and cold chain management to support any workflow. Now, sample mishandling, like poorly sealed products or leachable products, or even some temperature variation, can be detrimental to performance of a cellular product and can impact its overall quality. At Brooks Life Sciences, our consumables focuses on the quality and the traceability in that discovery space. Our Fluid X products can be dual-coated or tri-coated. This ensures sample tracking throughout the entire life cycle. And also, the Fluid X products easily fits into any automation pipeline. Brooks Life Sciences specifically has a comprehensive selection of instruments from single tube scanners to full rack readers, which can decode a full rack in less than 1 second, to full rack decappers and recappers, and also tube labelers. Now, our Fortitude products includes PCR consumables, sealing materials, and heat sealing instrumentation, microplates for cell screening, and storage, and solutions for sample tracking. And the sample tracking would include barcodes and 2D data matrix codes. The versatile portfolio really facilitates automation for reproducibility in any high throughput process. So when transporting samples within and even outside of the laboratory, it's critical to maintain cold temperatures to ensure sample integrity. For high-quality sample preparation, protection, and preservation, our BioStation range allows for reproducible results. This includes the Cool Cell, which offers alcohol-free controlled rate freezing at -1 degree Celsius a minute, the thermoconductive Cool Racks, which provides temperature uniformity across all vials when cooling, snap freezing, heating, or thawing, and the Cool Box, which keeps samples frozen or cold without ice. The Cryopad Carrier is a great solution that offers the ease of transport with the necessity of any temperature tracking. With customizable alarms and reports, this system provides a safe way to handle temperature-sensitive biological materials and minimizes concerns with sample integrity during the transit. So for cold storage, the BioStore 3 platform provides optimized cold chain and process control for a wide range of materials and critical reagents. This system includes an onboard software controller that documents and tracks user access, inventory, order history, and temperature trend reports. This is key for any high-value samples and reagents. This is also ideal where any situation that calls for an automated storage system and provides a comprehensive management system for any laboratory. Accordingly, Brooks offers off-site storage and management services or an on-site support with LIMS systems to help track and maintain any critical products that you may have. For off-site storage, we have many global repositories with the ability to track samples using a secure system, including role-based security and solutions that are FDA 21 CFR Part 11 compliant. For on-site management, we offer workflow and tracking using our Limfinity software. Now, Limfinity is easy to set up and also offers customizable features that allows researchers and laboratories to tailor the system to fit their specific needs. Next, I want to move on to the support that we have for clinical trials. Now, cell and gene therapies have brought a new paradigm to clinical trials. Traditional drug therapies have established regulatory pathways that take a compound or a molecule biologic through each stage from development to phase 3 trials in a very standardized manner. Typically, the number of patients grow from the tens in phase 1 to the thousands in phase 3. And once approved, these drugs are then targeted to treat tens of thousands, hundreds of thousands, or even millions of patients. Cell and gene therapies, on the other hand, tend to be a more targeted population, where traditional drugs have been ineffective. The trial sizes tend to be much smaller, with patients in the single digits at the phase 1 and only in the hundreds at phase 3. It's also common for phases to be combined for trials, for example, 1 and 2 or 2 and 3, as the patients tend to be very ill and are out of the traditional therapy options. Brooks supports advanced therapies at the preclinical and clinical level in multiple stages. So we discussed quite a few options under our client lab operations, such as providing the controlled consumables, the reagent management and storage, and the analytical and production services. For our material management solutions, we have the ability to have 24/7 visibility and access to the samples and products, regardless of where they are across the globe. And with the focus on sample and material management, we provide rapid retrievals of up to 1,000 items within 24 hours, so you can always get the right material to the right person at the right time. So whether that's a researcher that's using a sample to measure the effectiveness of a therapy, or if it's for a patient that's awaiting for their personalized therapy. For preclinical analysis, variables can be the root cause of irreproducible data. Now, Brooks quality processes ensure that all samples are handled in a consistent manner and in a way that minimizes sample variability. Whether it's through the use of our temperature-controlled work-in-progress carts or our expert staff that's extensively trained in the proper handling of any sample types, our staff ensures that samples will be handled with the utmost care and with the greatest attention to detail. In addition, Brooks is able to provide quality control assays in-house, whether it's verifying that the transfection process was effective or verifying the identity of a cell line or product. All of these offerings applies to any phase of the trial, whether it be a small size or a larger population study. All of our solutions are easily scalable and can be performed for research use or under CAP and CLIA compliant workflows. Further, Brooks has inline processing laboratories that use the same process across the globe to ensure that your assays are reflective of true study results and not the variation you may see between different processing laboratories. From basic fractionation to PBMC isolation, for cryopreservation and nucleic acid extraction, through genomic and transcriptomic analyses, our processes are well established to minimize any variables. Lastly, I'd now like to spotlight some of our supply chain solutions in the cell and gene therapy delivery space. Several unique logistical challenges arise with cell and gene therapy delivery. So it's really important to identify the right partner who offers flexible solutions to safeguard the integrity and quality of your drug product while meeting regulatory requirements and mitigating supply chain risks. When it comes to advanced therapies, accounting for the uniqueness of the product and the scalability of the process remains a concern. These therapies can be inherently unique. They are usually very high value, and in some therapies, like allogenic therapies, it's often produced in small lots. Autologous therapies are individualized down to a single patient, and the product often requires a unique storage need. Scaling up protocols to address all of these unique needs often requires complex and time-critical processes, and it often needs a significant investment. Most supply chains are also set up for warmer temperature storage, like traditional refrigeration temperatures, and sample tracking with chain of custody and chain of identity is very important for every single sample. At Brooks, we harness the strength of the N of 1 approach. This means that we treat every sample as an N of 1 and use the existing infrastructure built at Brooks and leverage our expertise in chain of custody tracking capabilities. So Brooks ensures safe and secure cold chain storage, transport, and chain of custody at every stage of the workflow, regardless of the therapy type. From sample collection to product manufacturing and distribution to end user, we have options to assist you at every stage. Our cold chain storage and inventory model can be categorized in 3 ways: storage at Brooks, storage on-site, and the hybrid storage model. The option that a company may choose is dependent on their life cycle and based on their overall capacity needs. When lower volume is the current source, complete outsourcing with Brooks might make the most sense. Then when a company is at a fuller scale, they might want to purchase and in-source more of their storage or go with a hybrid model. Now, for on-site and storage and self-management, we offer a variety of automated storage and inventory management systems, like the BioStore 3 that was mentioned earlier. For sites, this includes sponsors, contract manufacturing organizations, and logistic providers. We can also assist with storage at clinical sites. But regardless of the strategy chosen, any combination allows for real-time inventory visibility and data connectivity between all units and sites. All of our cold chain logistics solutions includes chain of custody and audit trail tracking. Robust solutions, it also includes some automated systems and a very fast turnaround. We have many licenses and accreditations that are listed on the right-hand side, and all of these accreditations apply to the logistics of distribution described on the left. So in summary, we've described several options as part of the cell and gene therapy workflow that facilitates a robust development pipeline. GENEWIZ AAV protocols can read, maintain, and prepare ITR samples with high quality and reliability. Our NGS capabilities allows for high throughput plasmid screening and provides AAV genome solutions beyond sequence confirmation. Brooks provides expertise in traceability, automation, and cold chain management through offering high-quality consumables, instruments, and cryogenic solutions via the FluidX and Fortitude consumables, BioCision products, BioStore platform, and Limfinity systems. And Brooks' supply chain offerings allows for cold chain logistics that reflect the unique nature of the samples, while continuing to provide a high level of sample accountability in a convenient, scalable, but customizable model. Now with that, I'm happy to take any questions. Thank you for your time.
Thank you, Dr. Louie, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you want to ask, please do so now. Just click on that Ask a Question box. And so let's get started. We have already several questions coming in. Dr. Louie, are there any limitations to the AAV-ITR Sanger sequencing service?
That's a great question. So yes, some recombinant AAV plasmids contain a homopolymeric G region that precedes the 5' end of the ITR region. So sequencing through this G region with existing Sanger methods have been very challenging, and even the ITR sequencing method suffers from that same difficulty. So what you'll see is that the signal will drop along the poly G sequence and causes a premature termination without extending that hairpin. So currently, unfortunately, there's really no good solution to overcome this hurdle. However, our AAV-ITR sequencing method is capable of sequencing from the reverse direction through that hairpin and that poly-C repeat. So it does improve the data quality and the read length in this region going from the other direction.
Thank you so much. Now, which Brook site is used to store patient samples from the clinical trials?
Yeah, so Brooks has 8 biobanks that are located on 3 continents, and those biobanks store clinical samples. So the primary locations, though, are in Indianapolis, in Indiana, in the U.S., Gresham in Germany and Europe, and also in Singapore and in Beijing, China.
Thank you so much. And again, I want to thank our audience for these great questions. Now, what kind of suitability do tubes and plates from Brook Life Sciences have for storing cell lines and PCR applications?
Yeah, so as I mentioned earlier, the Fluid X tubes is really great for cell line storage. So the tubes themselves are manufactured using a high-quality virgin polypropylene, and it's also manufactured in an ISO Class 8 cleanroom environment. So that means there's going to be no detectable leachables or extractables, and they're definitely endotoxin and DNase and RNase free. So it also has a great temperature range from -196 Celsius to 121, and that makes it really ideal for storing cell lines at a really cold temperature. But please note, it might not be the best for like a liquid nitrogen type of storage. When it comes to the PCR, the 4-2 plates are manufactured under ISO conditions as well, and we use the virgin medical grade polymers there. So the injection molding is done in an ISO Class 7 clean room, though. So it does ensure a high quality standards, and also, again, to make sure that they're free of any like leachables that can interfere with your PCR.
Thank you very much. And our next audience member wants to know, how does Brooks ensure appropriate inventory levels of critical reagents that are maintained?
So I assume this is referring to the biomaterial storage component. So at the start of every new project that's kind of initiated, there's a Brooks project manager that works with the clients to define what material is going to be stored. When that part is all set up, there can be alert and action limits that can be used as triggers. So there would be like an automatic reorder point notification.
Dr. Louie, we have time for one more question. Are you able to generate ITR mutants for efficacy testing and screening?
So we do have capabilities to perform de novo synthesis of any of the ITRs, or we can use our ITR correction protocols to mutate them. So the method that we use will be highly dependent on the mutant type that's being requested, but we do have a lot of experience just making entirely new AAV vectors and the vector backbones completely de novo. So we're happy to kind of discuss what options you are specifically interested in.
Dr. Louie, thank you for your time today, for your important research and your presentation. And before we go, I want to thank our audience for joining us today and for their interesting questions. And I want you to know that this webcast will now be available on demand through April of 2022. We hope you enjoy the event. Take care and stay healthy. Bye-bye, everyone.